besides using gel electropheresis to look for or diagnose diseases or screen for diseases, the most common use, the one that you see the most on CSI, is to use for forensics. So people have differences in their DNA, and if you cut them with restriction enzymes, because of those differences in the DNA, you'll get different banding patterns. So if you sort of see it in this gel here, this is the DNA or the banding pattern. So they found DNA at a crime scene and they cut it with restriction enzymes. And this is the banding pattern left by the DNA found at the crime scene. Same thing here with the DNA found from the victim. And you can sort of see these are the suspects. Here's suspect 1, here's suspect 2, and here's suspect 3. And you see that the banding pattern of suspect 2 matches up with the DNA sample found at the crime scene. And none of the suspects, and it's not the victim's DNA because the victim's banding pattern doesn't match up. So from this evidence, you know that at least, depending upon what the substance was that you got the DNA from, that at least suspect 2 was at the crime scene. Now the question is, why? What leaves this banding pattern? So like why do we have different bandings if we cut our DNA with restriction enzymes? This is actually, before we talk about that, this is actually a gel from a real sort of crime scene, and here is the defendant's blood. Here. So this is the person that is being charged with the crime. And then this is blood found on the defendant's clothing, and you can see that the banding pattern for the blood found on the defendant's clothing matches the victim's blood and doesn't match the defendant's blood. So the blood that he has on his clothing is that of the victim's. Now the question, and once again I'll say it again, the question that we're trying to solve is why when we cut people's DNA with restriction enzymes, do we get different banding patterns? And that's what we're going to talk about now. So if you remember back before Christmas, we were talking about sort of our DNA genome, and that a lot of the DNA doesn't code for anything. We have a lot of DNA that doesn't code for proteins. So in our DNA, we have these sections of sort of, and actually this is sort of becoming a different, like, of what they call junk DNA, but they're actually finding that some of this junk DNA actually does stuff. But that's not for today's lesson. So we have these areas of DNA that don't code for anything. They don't code for proteins. And they're made up of many repeats. And because like this area of the genome doesn't code for proteins, it can have mutations in it, and nothing happens to the person. So normally if there's a mutation in a gene, a lot of times that causes the protein not to function, and the person either will die, or if it's a baby, it won't make it through development, and the mother will have a miscarriage. But mutations in junk DNA don't really bother the person because they don't code for anything. And these areas of junk are made up of many, many repeating sort of sequences of different lengths. So maybe this is the DNA of someone, and notice that they don't have as many repeats in this junk DNA as someone else. If you actually look at someone's DNA, human DNA, we're very, very similar, except for these, because we all have the hemoglobin gene. We all have cytochromes for gel electropheresis, for electron transport chain. So our DNA is very, very, very similar, except for these junk regions, which don't code for anything. And because they don't code for anything, they're more likely to get mutations in them that don't affect us. So if you look here, this is an example. The person down here, so allele 1, 2 different people, they don't have as many repeats as the person here in this section, in the same sections of DNA. So when you cut this DNA with restriction enzymes, it's going to leave a longer band. So if you sort of see, we look down at the gel at the bottom, too fast. Allele 2, because this is longer, this DNA strand here is longer where it cut, the restriction enzyme cut on either side here, the band on the gel electropheresis doesn't travel as far because this has longer base pairs, whereas this has shorter number of base pairs, so it travels further down the gel. These differences, you can try to say this 3 times fast, these differences people's DNA and the junk DNA are called restriction fragment length polymorphisms, or RFLPs for short, so RFLPs. Restriction, because we're cutting with restriction enzymes. Fragment, because we're making fragments. Length, because they're different lengths. And then poly means many differences. So, I mean, that's sort of where the word comes from. So we cut by restriction enzymes, then remaining fragments, and then we make fragments of many different lengths because of the differences in our junk DNA. And once again, here we can see the difference in banding pattern here. So in this person, they actually have, well, this person here, if you look at the alleles, the difference between the two alleles here, let's say this is in one person, this is in John, and this is in Mary. So here, because of a mutation in the junk DNA, they actually have one less cut that happens. So this person here, the restriction enzyme cuts twice, leaving 3 fragments. And here, the restriction enzyme only cuts once because of a mutation in the RIF flip, or the area of junk DNA. And then because of this mutation, and because of the difference in cutting, we get different banding patterns. Which it says here creates different fragment sizes based on the cutting up here. Okay, so once again, just to repeat, because we have differences in our junk DNA, and if you put restriction enzymes in our DNA, because of these differences, the restriction enzymes are going to cut differently because of the differences in the sequence. And as a result, because they cut differently, they give different size fragments. So here we have 2 fragments, therefore we get 2 bands on the gel. Here we only have 1 fragment and didn't cut, and then we get only 1 band on the gel. And then here we get it cut twice. I'm sorry, well, let's see. Yeah. But here, because the fragments are different sizes, we cut different places here, we get different banding patterns because of the differences in the sequences in the junk DNA. Because of these differences in sequences, you can tell whose DNA is who when you run it on a gel, because we get different banding patterns. If we look closer in here, at this is a one single base mutation that occurred right here. Because of that single base mutation, remember this is junk DNA, so the mutations don't hurt us. Okay, the restriction enzyme here doesn't cut anymore, and that will leave a different banding pattern. Here, because we had part of the DNA that was duplicated or replicated twice in the junk DNA, remember that doesn't hurt us in this case because it's junk DNA, then we'll get a longer banding pattern, which will be up closer to the gel because it's longer, therefore it doesn't travel as far. This picture here shows, in 1987, I was 2 years old, shows the first sort of gel that was used, or first time DNA evidence was used in a case. I mean, you can sort of see that the gels, they were very rudimentary at the time. They don't, you saw better pictures from previously, but you can sort of see that the semen found matches the DNA from the suspect. The banding pattern is matched. Obviously, the gels have gotten much better since then. But also the same sort of thing here. You see that the banding patterns match up. Obviously, the gels have gotten much, much better at this point. But Ralph over here, he's asking a question, how can you compare DNA from blood and DNA from semen? And if you remember, all cells pretty much have the same DNA. Semen cells have half the amount of DNA, which we'll talk about next week or the following week after the TIS. That's what we're going to get into. But remember, all cells have the same DNA, which just different is which genes are turned on and off. So this is sort of a more advanced gel. So you can see that this is the DNA found at the crime scene, and because of the differences in junk DNA, when you cut it with restriction enzymes, it leaves different RIF flips, or restriction fragment length polymorphisms. You can sort of see that the DNA found at the crime scene matches the banding pattern from suspect 2. The last use that we're going to talk about, I think, let me just see, yeah, the last use we're going to talk about is paternity, and it sort of uses the same sort of ideas that we use for forensic cases, except that the banding pattern is only going to match up for half, because half of the DNA comes from mom, and half of the DNA comes from dad. So once again, we're still using the same example of the restriction fragment length polymorphisms, that everyone has different ones. But in this case, the question is, who is the father? And half of the child's bands are going to come from her mother, and the other half are going to come from her father. So if you take a look here, let me get my pen back out. Pointer options. So some of the bands, so like this band right here comes from mom, and this band right here also comes from mom. But the question is which father? do the other half of the bands come from. And you can see that the father 2 here matches up at some of the bands here, whereas father 1 doesn't match up at hardly any of them. From using sort of the restriction length polymorphisms, you can see who the father is, who has the other half of the banding pattern from the child. From that, using the same sort of differences in junk DNA, and then because of the differences in junk DNA, the restriction enzymes cut at different spots, and that causes a different banding pattern. All right, so that is the end.